These cinefilm sequences were recorded using a Zeiss inverted microscope with a ×40 objective and a ×2.5 optivar in either phase or interference phase contrast, linked to a Canon digital camera and a Panasonic CD-RAM recorder. Windows Movie Maker was used to edit the film. The width of view at the largest magnification on the TV screen is 120 microns. I used soil from my garden at Viewfield Midmar for these experiments. Large boiling tubes were filled with this soil to a depth of 3 centimeters, and microscope slides were placed on top of these soil samples. The boiling tubes were then filled with sufficient of Hoagland's plant nutrient solution to cover most of the area of the slides. Plant seedlings or root segments of Scots pine, that's Pinus sylvestris, or white clover, Trifolium repens cultivar Huia, were attached to the slides with Teflon tape. The plant stems and leaves were arranged so that they were just above the nutrient solution. The boiling tubes were incubated at 20 degrees centigrade, either in the dark or in sunlight. Obviously, the conditions on roots in soil are more complicated than recorded here, but this technique does illustrate the range of protozoa present on the root surface and some of their interactions. Protozoan activity on the root surface is most intense near the root apices and where lateral roots emerge from the main roots. Here you can see one of these boiling tubes with a microscope slide and plant seedlings on top of the soil sample. This is a root apex of white clover seedling, and you can see large numbers of flagellates moving around the root cap cells. Later, at greater magnification, you will see a Limax soil amoeba moving along the surface of the root cap cells before finally moving in amongst the root cap cells themselves. Clearly, the root apices are centers of intense protozoan activity. first example of soil flagellates is a small Circomonas species with two flagella, one pointing forwards and the other trailing behind. Note the amoeboid extension at the back where bacteria are being engulfed. There are many species of Circomonas in the soil. Most are larger than this specimen. This body is about 8 microns long. The second example is a Heteromita species of about 10 microns long with a short active flagellum and a longer posterior flagellum. The anterior flagellum creates water currents that bring bacteria and other food particles to the front of the flagellate, where they are engulfed by a temporary pseudopodium. Pusomonas proboscidea is the third flagellate. It has an unusual flexible proboscis or trunk to which two flagella are attached, one pointing forwards and the other backwards. This flagellate characteristically moves along the edges of soil particles and root surfaces, eating large numbers of bacteria. The next example is a flagellate enclosed in a transparent organic lorica on a root. This is a species of Bicozoica with two flagella, a long one pointing forwards and the other one is attached to the bottom of the lorica. The lorica is about 10 microns wide. Both flagella can contract at intervals, and as a result the cell is drawn down to the bottom of the lorica. Later the flagella relax and the front flagellum is thrown forwards again to catch more food bacteria. Here we have 4 flagellates of Spumella species on a root hair. Each flagellate has 2 flagella, a long one that creates the water currents and a shorter one that helps to incorporate the fruit prey into the front of the body with the help of a short pseudopodium. Some Spumella species are free-moving and not attached. Monozygous species has a single flagellum inside a collar at the anterior end of the body, and it is attached to a root by a stalk. This flagellate often occurs in colonies. Spongomonas species also often occurs in colonies. It has 2 relatively long flagella on the left and is surrounded by a capsule of mucilage with attendant bacteria. Now we are amongst the soil amoebae. This specimen has been floating in the soil solution and has assumed a form that is difficult to identify. It is green because it has been eating algae. It is small and slow moving, and thus conforms to the popular view of soil amoebae. The next amoeba is a species of sac amoeba and is clearly moving at much more rapidly than the previous one. It is monopodial. You can see a central nucleus, a posterior contractile vacuole, and many refractile bodies in the protoplasm. Here we have a larger amoeba, a species of Mayarella. Instead of a monopodial pseudopodium, it has several finger-like pseudopodia. If you watch the posterior part of the body, you will see that this amoeba excretes a group of particles that subsequently stay together and do not readily disperse. It is possible that these particles are enclosed in a membrane. If this assumption is correct, it may have important consequences for further decomposition. The rapid movement of many particles within this amoeba is a good demonstration of the living nature of this organism. This is a giant amoeba, Arachnula impatiens. It can reach up to 1 millimeter in diameter. It has many nuclei and many contractile vacuoles. This amoeba can produce large quantities of mucilage that can trap suitable prey for subsequent engulfment by the fine pseudopodia. It belongs to the family Vampyrellidae. It can cut holes in many fungal spores and remove the cell contents, so it is aptly named. It is also known to attack plant pathogenic nematodes, but it is not known if these amoebae can control nematode or fungal populations in the soil. At the end of this sequence, you will see in the lower part of view some pseudopodia pulling apart. And these amoebae can fragment into several separate entities. It can also reproduce in division cysts, where all the nuclei divide simultaneously.